Hi, this is Dr. Arpitansu, and today we're going to talk about the complement system. This is a part of immune system, and these system comprise of 20 or more than 20 proteins, which are generally inactive in your blood. But whenever there is an inflammation or any pathogen introduction in your blood, they gets activated uh, by certain proteases or any stimulating factor. Okay, and they augment the immunological reactions, help in inflammation, and also damages the cell membrane of the pathogen. Okay, let's see what is the complement system now. Let's begin with the introduction. The complement system is also known as the complement cascade, and this cascade comprises of more than 20 proteins. These are named as C1, C2, C3, and so on. And it is a part of immune system that enhances the ability of antibodies and phagocytic cells like neutrophils and macrophage cells to clear the microbes through opsonization, maybe direct opsonization or indirect opsonization, and damage cells from an organism to promote the inflammation and attack the pathogen cell membrane. It kills the cell membrane of the pathogen, or it creates the perforations or pores in it, and these pores causes the disturbance in the internal environment of the pathogen, and ultimately the cell dies. Okay, it is a part of innate immune system, and which is not adaptable and does not change during an individual's lifetime. The complement system can, however, be recruited and brought into action by antibodies generated by the adaptive immune system. Now, this complement system comprises of small proteins and that are synthesized by the liver. And these small proteins C1, C2, C3, and these all are synthesized in your liver hepatocytes, and they circulate in the blood as the inactive precursors, and they don't get activated until unless there is an inflammation or there is a stimulating signal to them. And when they get stimulated, the proteases in the system cleave the specific protein to release cytokines and initiate an amplifying cascade of the further cleavage. For example, first the C1 gets attached to the antigen like IgG or IgM, and then C1 activates the other C4, then C2, then C3, and so on. The cascade is formed. Okay. Now, the end result of all these complement activation or the complement fixation cascade is stimulation of phagocytes to clear the foreign and damaged material, or inflammation to attract the additional phagocytes, and activation of the cell killing membrane attack complex. Now, these membrane attack complex called as the MAC, these are comprised of the last C5, C6, C7, C8, and C9 complement proteins. Okay, and over 30 proteins and protein fragments make up the complement system. And there are three biochemical pathways which activate the complement system. We will talk about that today. The first one is the classical complement pathway. Second is the alternate complement pathway. And the last is the lectin pathway, or also called as the MBL pathway, which is the mannose bound lectin pathway. Okay. So let's start with the classical pathway. Let's see what is the classical pathway. This pathway involves the complement C1, C2, and C4, and the pathway is triggered by the antibody antigen complexes, which is the which is the characteristic of this pathway, binding to the C1, which itself has three subcomponents: C1q, C1r, and C1s. And the pathway forms a C3 convertase, C4b2a, which splits C3 into two fragments. The larger fragment, C3b, can covalently attach to the surface of the microbial pathogen and opsonize them. The small fragment C3a activate the mast cell, causing the release of the vasoactive mediators such as the histamine. Now, this paragraph has a lot of words. For example, we have a C1, C2, and C3. These are the complement proteins. Okay? These are the proteins which was synthesized in your hepatocytes in the liver, and they were inactive. But once they get any stimulation, for example, IgG and IgM, they bound to the antigen present on these IgG and IgM antibody. When they bind to the antigen present on the pathogen, they the Fc chain of the IgG or IgM uh, activate the C1 complement protein. Okay, and this C1 complement protein has three legs, and these legs are C1q, C1r, and C1s. And the C1q leg of the complement 1 bind to the IgG or IgM, and it further amplify the cascade by activating C4b, C4-2b, and so on. And these C4b and C2b, These are called as the C3 convertase, okay? And these activate the C3, and then C3 further gets cleaved into C3b and C3a, okay? And the C3a is a strong anaphylatoxin. You should remember that. And it also is one of the opsonizing agent. And C3a causes the indirect opsonization, okay? And yeah, so this is how the classical pathway work. Classical pathway is basically few points to be remembered, that it is antibody antigen complexes, that in the classical pathway it is important that at first IgG or IgM, only these two immunoglobulins should have to be bound on the antigen on the pathogen, okay. Secondly, the first complement protein which gets activated is the C1, okay. And these was the two characteristics of the classical pathway. Now let's see the lytic pathway. Now once C3b gets activated and C3a separate from the C3 and C3b bind to the C2a, C3b then activate the C5, which further gets cleaved into C5b and C2a. Now this C5b, now to this C5b binds the C6 and C7, and then C7 and C8. Now this in last tail of this whole complement cascade, that is C5b, C6, C7, C8 and C9, they all form the membrane attack complex, and these bind to the cell membrane of the pathogen and create pores in it. Okay, so let's see. So this C4b, C2b and C3b, these all three combinedly called as the C5 convertase. You should remember what is C3 convertase and what is C5 convertase. So when I am talking about C3 convertase, it is only C4 2b and C2b. But when I'm talking about the C3 convertase, another protein C3b is connected to the C2b. Okay, so the whole C4b, C2b and C3b except C1. C1 doesn't have any B or A part. You should remember that. C1 is a whole protein. Okay, and this C4b, C2b and C3b, they are called as C5 convertase, and it splits C5 into C5b and C5a. C5a is a potent anaphylatoxin, and it can go up to the mast cell, and it can go up to the mast cells or any other cells, and generally it causes other inflammatory cells to come into the area where the whole accident is happening. Okay, now to this complement, to the C5 complement, C5b complement, C6 and C7 bind, and then we have C8 and C9 which bind, and ultimately the C9 creates the pore in the cell membrane of the pathogen and causes the internal environment damage of the cell, and the cell further gets apostatized. Now over here we have a diagram of a normal antibody. You can see that this is the heavy chain of the antibody and these are the light chains. Okay, now over the this part, we call this part as the FC chain, and in the FC chain you will see that there is a region. We call this region as CH2 region. Now, this CH2 region is a complement binding site, and whenever the complement number 1 comes into the action in the classical pathway, it binds to this CH2 site of the antibody, this complement binding site. You should remember that. Okay, so let's uh, let's revise once again. This is a diagram of the antibody. Okay, and this is the site of the antigen binding site. It binds with this site. Okay, this site binds to the antigen which was present on the pathogen, and these are the heavy chains, these are the light chains which are bind together by the disulfide bonds over here. Okay, you can see this disulfide bond, and this is the FC region of the antibody, and on this FC region we have CH2, which is the complement binding site, and over this complement binding site the C1q bound. Okay, now let's see uh, the structure of C1, the classical component pathway, the complement number 1 protein. Now this complement 1, as we know, has 3 legs: C1q, C13, C1q, C1r and C1s. Now over here you can see that this blue bundle is C1q, and it has 6 globular legs. Now these, you can see the 6 globular legs it has, and it also has C1s and C1r. Okay, and these C1s and C1r are the the both are the serine protease. Okay, these are the serine protease, and the total number of C1q is, there is only one molecule of C1q, but there are two molecules of C1s and C1r. So the whole structure of a C1 complement can be summarized as C1q r whole square s whole square. Now over here you can see the MBL lectin pathway. The MBL, which is the mannose binding lectin, it also has a structure which is almost similar to the C1. Okay, it also has MBL, which is one molecule of MBL, which is similar to the C1q, and MASP2, which is the mannose associated serine protease, which is similar to the C1s protease, C1s serine protease, and MASP2, that is the mannose associated serine protease 1, it is similar to the C1r serine proteases. Okay, so this was the structure of C1 protein. Now over here you can see that these IgG, when they bound to the surface of pathogen, the C1q of the C1 complement, its globular head bind to the CH2 region of the IgG's Fc body. Okay, so over here you can see that the globular head of the C1q bind to the CH2 region in the Fc chain of the IgG antibody, which was binding to the antigen on the pathogen. Okay, so you should also see that there are 6 globular protein. Okay, so therefore there should be 6 IgG. You should remember that there should be 6 IgG to activate the complement pathway. Okay, so that each of these 6 globular uh, region of the C1q molecule should bind to the 6 IgG body. But contrast to that, only one IgM can activate the complement pathway. Now over here you can see that there is a IgG whole 6, that there are 6 IgG bodies, and this is a C1 protein, and these are binding to the, okay, so complement initiation determinants, you can oversee it. Now over here you can see the C1s and C1r, they are getting activated. Now whenever the C1q's globular part attaches to the Fc chain of the IgG, that is the CH2 region of the Fc chain of the IgG, they activate the C1s and C1r because there is a conformational change in the C1q. Okay, so whenever they bind, whenever the C1q bind to the antigen, it brings a conformational change in it, and this conformational change in C1q activate the C1s and C1r. Okay, now in and these C1s and C1r, they are the serine proteases, you should remember that. Let's see. So over here you can, I have summarized the whole classical pathway. At first the C1q bind to the antigen, and then this C1q activate the C4b, then C2b, then C3b, and then C5b, and on C5b, C6, C7, C8 bind, and C9 creates the pore in the bacterial membrane. So you can see it again, that first the antigen are bound to the bacteria, and then the C1q protein first attached to it, then comes the C4b, then comes the C2b, then comes the C3b, and then C5, and C5 attaches C6, C7, C8, and C9, and the C9 creates the perforation in the cell membrane. Now whenever these attach, okay, you should remember that C4b, only the B part of the complement protein bind to the surface, and the A part gets separated away. Okay, so B for binding and A for away, you should remember like that. And over here you can see the on the cells of the pathogen, complement component C5 to C9 assemble a complex that perforates the cell membrane. So you can see that this is C5b, and this to this C5b, first C6 and C7 attach, and then C9, C8 attach, and then C9 creates a pore in the cell membrane. Okay, now to prevent this on the RBCs and other cells of the body of the human, there is a certain regulatory endogenous protein present on the surface. Now these regulatory endogenous proteins such as the CD59, which is present, and CD59, it prevents the C9 polymorphism to occur, so that there is a prevention of pores to be created on the self cell. Okay, so here you can see the CD59, which is an endogenous regulatory protein. So there are other endogenous regulatory proteins like CD55, CD46, and CD35. Okay, so this CD59 prevent what? These CD59 prevent the C9 polymorphism, so that they can so there is no pore creation in the self cell. And over here you can see these how these pores are formed. This is the cell membrane, and these are the complement protein which created the pores, and the extracellular fluid get flushed into the membrane, and the internal environment of the cell get disturbed, and the whole cell dies. Now let's talk about the second pathway, which is the alternate pathway. Now this pathway involves various factors like B, D, H, and I, and which interact with each other and with C3b to form a C3 convertase, C3b BB, that can activate more C3. Hence the pathway is sometimes called as the amplification loop. The activation of the loop is promoted in the presence of bacterial and fungal cell wall, but is inhibited by the molecules on the surface of the normal mammalian cell. Okay, but the alternate pathway is different from classical pathway, because it doesn't require any antibody to be attached to the antigen of the pathogen. So it is independent of the antigen antibody reactions. Okay, and there are certain factors like there's B factor, D factor, H factor, I factor. We will talk about it. These are involved in the alternate pathway. Okay, so these alternate pathway occur at the low level. Okay, and it begins because of the hydrolysis of C3 into C3b and C3a, and it occurs at the low level, and this hydrolysis occurs due to the breakdown of internal thioester bond of C3. Okay, so this is a spontaneous hydrolysis. At first, the first step of the alternate pathway is that there is a spontaneous hydrolysis, and this spontaneous hydrolysis break down the internal thioester bond of the C3 into C3b and C3a. Now you should remember that in the fluid state, the factor H, this factor H inactivate the C3b in the fluid stage. But when the C3b attaches to any pathogen, it is protected from the factor H, and then comes the role of the factor B. The factor B then binds to the C3b, and factor D cleaves the B into BB and BA. Now this C3b and BB in the alternate pathway act as a C3 convertase. Remember, in classical pathway, the C3 convertase was C4-2B and C4B and C2B. But over here, the C3 convertase is C3B and BB. Okay. Now this C3B and BB act as a C3 convertase and then recruit another C3, which gets cleaved into C3B. Now this C3B BB, you should also remember that there also comes a factor called as the factor P. We call it as propedrin. Now this propedrin, what it does is that it stabilizes the C3b and BB. Okay, so factor P called as the propedrin, it stabilizes the C3b and BB, so that the C3 convertase get stabilized, and after the stabilization, the C3 more C3 gets activated. Okay, more C3 gets activated. Now, after that, we know all the steps that after C3b gets activated, then it activates the C5 and all the MAC complement or any opsonizing part of the opsonizing. Okay, obviously C3b is itself an indirect opsonizer. So it can either kill the cell by opsonization, or it can also form the membrane attack complex. Okay, so this pathway is sometimes called as the amplification loop, because you can see that activation of the loop is promoted in the cell upsurface of bacteria and fungal cell wall, but is inhibited in the molecules on the surface of the normal mammalian cell. So you should remember how it is different from the classical pathway. It is different from the classical pathway because it doesn't because it is independent of the antigen antibody reactions, because classical pathway was dependent on the IgG and IgM. Okay, and but it is independent of that. It can in this pathway, the C3b can directly bind to the pathogen. Okay, and after binding, the it doesn't get by after binding to the it doesn't activate the C5 directly. At first, to the C3b, the B factor bind, and this B factor then gets cleaved into BA and BB, and then this C3b and BB gets stabilized by the factor P, which is the propedrin, and this propedrin stabilizes the C3b and BB, or we can say the C3b or BB as C3 convertase, and more C3 gets come into the axon, and further the opsonization occur, or the or membrane attack complexes are formed. Okay, so over here you can see that first the C3 was spontaneously hydrolyzed, okay, by the breakdown of the internal thioester bond. Then the C3 convertase, over here you can see, okay, C3, the C3 convertase, now in normally C3 convertase and these C3 they get inactivated in the fluid stage because of the factor H, but once they get bind to the pathogen, they get activated and they get protected from the factor H. Now, okay, you can see that C3b, when it's bind, then factor B attaches to it, and the FT, factor D, cleaves the factor B. Okay, so the factor D cleaves the factor B into BB and BA. Now, to at this C3b, the BB is attached, and this is formed as the C3 convertase. Okay, so it goes and convert more C3 into C3b and recruit more C3. Okay, and once C3b is again attached to this BB, then this whole C3b, BB and C3b uh, complex is called as a C5 convertase, and then it recruits the C5, and then it gets uh, split into the C5b or C5a, and then MAC, that is the membrane attack complex, are formed. Okay, so let's revise it again, that at first there is a spontaneous hydrolysis due to the breakdown of the internal thioester bond of the C3, and in the fluid stage this breakdown and this C3b activation was inhibited by the factor H, but once it get attached to the cell, it is protected from the factor H or I, the two factors, factor H and factor I. So once it attaches to the pathogen or the cell, it gets protected from the factor H and factor I. And to this C3b, first the factor B gets attached, and this factor B is cleaved by the factor D. So once to the C3b, the factor B was attached, and this factor B was cleaved by FD or the factor D into BB and BA. Now this BB attached to the C3b forms the C3 convertase, which recruit more C3 to the cell, and once one more C3 when attached to this BB, then the whole complex C3b, BB and C3b is called as a C5 convertase, and then it splits the C5 into C5b and C5a, and then whole the membrane attack complex that we have already studied is formed, and it creates the pores and all that, this all the things occurs. Okay, now let's talk about the last pathway, which is the mannose binding lectin pathway. Okay, so mannose binding lectin pathway is activated by the binding of the mannose binding lectin, MBL. We have seen that this mannose binding lectin has a structure which was similar to which complementary protein? It was similar to the uh, complement number 1 protein. Okay, so this pathway is activated by the binding of the uh, mannose binding lectin to the mannose residues on the pathogen surface. This in turn activates the MBL associated serine protein, that is MASP1 and MASP2, which activate the C4 and C2 to form the C3 convertase and convertase C4B2A. So it is much similar to this classical pathway, but the only difference is that instead of C1 protein, C1 complement protein, over here we have the mannose binding lectin, MBL, which is homologous to the C1 complementary protein in the structure, as it also have one molecule of the mannose binding lectin, which is similar to the C1q of the C1, and it has two serine proteases, that is the MASP1 and MASP2, that is mannose associated serine protease 1 and MASP serine, and mannose associated serine proteases 2, just like the C1R and C1S of the C1 molecule. And in these in turn activate the C4 and C2, and then split it into C4b and C2b, and further the next steps are similar to the classical pathway, that this C4b and C2b act as the C3 convertase, and it recruits the C3, and then C3 recruits C5b, C5, and then it splits into C5b and C5 2a, C5a, C5b and C5a, and then C5 recruits other C6, C7, C8, and C9, and the further MAC is formed. So the mannose binding lectin pathway is much similar to the uh, classical pathway, with the only difference that over here the first complement protein, that is so first protein which is binding is the manose binding lactine, okay, which is homologous to the structure of the C1 with three legs. First, the if three legs, that, and also the two serine proteases like MASP and MASP2, which is similar to the C1R and C1S. Over here you can see that this MBL is similar to the C1Q with six globular heads, and then two serine proteases C1S and C1R. Over here you can see the manose serine proteases 2 and manose associated serine proteases 1. Now the regulation. Okay, so coming to the regulation, so there are certain endogenous regulatory factors also, so that these complement system doesn't kill our own cells. Okay, so to regulate these, there are certain factors. We have already talked about CD59 previously. Okay, as the CD59 is also called as the protactin. Okay, so this CD59 also is called as the protactin, and it inhibits the C9 polymerization. We have already seen that it inhibits the C9 polymerization, and so that the uh, pores are not created on the cells. Okay, and these are present only on the cells, and these are not present on the pathogens. Second, we have C1 inhibitor. Okay, so C1 inhibitors are so we have C1 inhibitor, and third we have decay accelerating factors. Now these decay accelerating factors are the GIP on the red blood cells. Apart from these three, the CD59, C1 inhibitor. Okay, so D1 DIF, decay accelerating factor, it inhibits the C3 convertase. Okay, so DIF, that is decay accelerating factor, inhibits the C3 convertase, and these are the GIP on the red blood cells. CD59, what does it do? It inhibits the C9 polymerization so that the pores are not created on the cells. And we have C1 inhibitor. Okay, and we have other endogenous regulatory factors such as the CD55 and CD35, CD46, and many more. Now let's coming to the diseases. Okay, so let's first begin with the deficiency diseases. Okay, so the diseases are systemic lupus erythematosus, glomerulonephritis, various form of arthritis, autoimmune heart diseases, multiple sclerosis, inflammatory bowel syndrome, that is IBS, paroxysmal nocturnal hemoglobinuria. And this paroxysmal nocturnal hemoglobinuria is basically because of the deficiency of C1 inhibitor. And we also have atypical hemolytic uremic syndrome, and deficiency of the terminal pathway predisposed to the autoimmune diseases, mainly the Neisseria, gonorrhea type of diseases. Okay, now mutation. Okay, so over here you can see that I have written that it plays particularly Neisseria meningitidis due to the role that the membrane attack complex plays in attacking the gram-negative bacteria. So gram-negative bacteria are more vulnerable to the MAC complex than gram-positive, because gram-negative bacteria has a what do we say? It has an outer lipid layer which is more open to the MAC complex as compared to the thick layer of the glycoprotein of the gram-positive bacteria. Okay. Now mutations in the C1 inhibitor gene can cause the hereditary angioedema, a genetic condition resulting from the reduced regulation of bradykinin by C1-INH. So thank you. We will end this chapter. Let's meet again.